Good evening, everyone, and welcome to the Fralin Biomedical Research Institute at VTC's Maury Strauss Distinguished Public Lecture Series. Hope everybody is staying well. It's good to see you out there and look forward to a great evening tonight. Before I have the opportunity of introducing tonight's speaker, I want to give a shout out and an advertisement for the next talk coming up next month. And that will be a lecture by Dr. Michael Young, who is at the Rockefeller University in New York. Dr. Young will be talking about genes regulating sleep and circadian rhythms. Dr. Young is the Richard and Jean Fisher Professor and Vice President for Academic Affairs at Rockefeller. He's also the recipient of the Nobel Prize in Physiology or Medicine a few years ago for his seminal work in this area of regulating sleep and circadian rhythms. So we hope many of you will join us for the next lecture that'll be coming up on March the 25th. So without further ado, it is indeed my great pleasure and honor to introduce this evening's speaker. And before I get into the introduction, I want to mention to everybody, hopefully when you signed on, you saw a poll pop up on your monitor. And that poll has one question, which is, if you had the opportunity to take a COVID-19 vaccine next week, would you take it? Yes or no, or not sure. And it is optional. You don't have to fill it out, but for those of you interested, please do. And our speaker, Dr. Fuller, will see the results. If it hasn't popped up on your screen, if you click on the bottom on the taskbar at a little icon that says polls, it should pop up for you then. Okay, so it is indeed my great pleasure this evening to introduce our speaker, Dr. Oveta Fuller from the University of Michigan. Dr. Fuller did her undergraduate studies in biology in her home state of North Carolina at the University of North Carolina. She then went on and completed her Ph.D. in microbiology and immunology, also at the University of North Carolina. And then she did postdoctoral training in molecular genetics and cell biology at the University of Chicago, where she also later was appointed to the faculty. And then in 1988, Dr. Fuller joined the faculty at the University of Michigan in the Department of Microbiology and Immunology. She served in a variety of leadership roles there, in addition to being on the faculty, including having served as the Associate Director of the African Studies Center at the University of Michigan. Dr. Fuller has been recognized in numerous ways for her many contributions. She's served on numerous study sections or review panels, the National Institutes of Health and the National Science Foundation, evaluating grant applications for doing research, for example. She's a founding member of the Bioethics Commission of the Tuskegee University Center for Bioethics, a very important center, and played an important role in that particular organization. She also recently was appointed as a member of the Vaccines and Biologic Products Advisory Committee for the Center for Biologic Evaluation and Research at the FDA, the Food and Drug Administration. And that has direct bearing on tonight's topic, as you'll hear. In addition, she's been involved as a Fulbright faculty scholar for the Sub-Saharan African District, and where she also won a Distinguished Service Award in Microbiology and Ministry from the AME Church. And speaking of the AME Church, a highly, I would say, rare condition for people that are very successful in science to also be an ordained minister in a church, formerly as a pastor at the AME Church. And she is an elder in that church as well. And so through that, Dr. Fuller brings an incredible perspective. Her studies and work over the years dealing with infectious diseases and pandemics related to HIV and AIDS, for example, also influenza and other diseases, most recently related, of course, to COVID-19, have put her in a position where she has the incredible scientific credentials as a virologist who has done work on and published leading edge scientific research in her understanding of infectious diseases, but also has then translated that and connected that to how that information is accurately and faithfully disseminated to communities, to help communities to be able to use that information for making informed decisions, whether it's about certain behaviors, health behaviors, for example, or vaccinations as it may be. And as we all know, living through this particular age in the pandemic, we have a lot of challenges in front of us right now. Not only the challenges of identifying this virus and the various mutations and variants, but also the challenge, of course, of coming up with appropriate ways through health behaviors, working with health departments, and also vaccinations to mitigate the effects that are often severe and deleterious. But that's only part of the story, right? A big part of the story is getting us, the public, to understand and accept and be able to use that information to make the best and most informed decisions on our own behalf and the behalf of our families and our communities. And it really takes people like Dr. Fuller, who are not only expert, but have dedicated their lives, their passion, their intellect, and their work ethic to bringing this information to communities, in many cases underserved communities, and then tying in through the faith communities as well with churches, both here in the United States and in Africa, in Dr. Fuller's case, to help all of us have the best information to help our health. So it is truly a timely visit, of course, with all that's going on by Dr. Fuller, and a real great honor and pleasure to have somebody here of her caliber to tell us this evening about trusted messengers and being able to address this particular pandemic in terms of COVID-19 and vaccination. So without further ado, I will turn it over now to Dr. Fuller. Welcome, Dr. Fuller.
Thank you so much, Dr. Friedland. It's a joy to be here. I appreciate the invitation, especially for this Maurice Strauss Distinguished Public Lecture. And so I'm grateful for this time to share with you. It's been a wonderful day meeting faculty and students. And so what I'd like to do, hopefully everyone was able to finish the poll and give us your thoughts on these matters. I'm going to now share my screen. And so we'll get started. It's really an honor to be here today. And I hope that before the hour and a half is over, we will have made some progress in getting some understanding. When I was asked to come to the Fralin Institute, I thought I was going to be talking about trusted messenger, but as time evolved, I realized that perhaps the more useful thing would be not to talk about the research with HIV and development of trusted messenger, but perspectives on coronavirus, COVID-19, and vaccines as we all navigate this pandemic together. So what I do want to do is pay homage to the work that we're still doing, but now with COVID-19. And that's the idea of the trusted messenger, fulfilling a need where we use religious leader networks to reframe conceptual community perceptions about disease prevention. So whereas it's been worked out with HIV, it could be used with anything that needs to go from the laboratory and clinics to people for implementation. So the strategy uses in-depth science-focused education with networks of religious leaders to effectively and confidently address whatever the issue is. And I just wanted to show that I'm not doing this all by myself. Over the years from 2007 to now, there have been a plethora of people and support, as well as now a program called the MQ program at the University of Michigan. So I also like to give people the takeaways beforehand so they can know what they're going to be hearing about and make sure that they listen for those things. And that way I can be very successful in the seminars because I tell you what I want you to know so you know it. So the takeaways today are threefold. One, that pandemics are part of adaptable nature. Human, animal, and environmental contact, as they occur more and more, the whole world was made in a way that it is adaptable. And so what we're seeing is adaptation of a virus from one host to another. The second thing is that for a COVID-19 vaccine, which is what is available now, you must make an informed decision. Nobody can make that except you and your family for each of you. But at that 95% effectiveness, and I'll explain what that means, in preventing COVID-19 disease, even for a little while, a COVID-19 vaccine is a great tool in our toolkit to help us manage COVID-19. We are going to coexist with this virus just like we coexist with influenza. And so this will help us get there where we can get back to a life of being able to hug our grandkids and meet with our families and enjoy the things that allow us as social beings to be human. And then the third point is whether you get a vaccine or not, you must make and implement an infection prevention plan. And that plan must include the things that we know work well to stop viruses, particularly respiratory viruses. And that's wearing a mask, keeping a certain distance, and I'll explain that, not gathering outside of your family unit until it's safe to do so, hand washing to get rid of any virus that may be on your hands before you touch your face. These things are absolutely necessary and will be for some time to come, even as we roll out immunity from either the vaccine or the disease. We should not be gathering outside of our own families. We should not really be traveling unless it's an absolute emergency. And then lastly, we need to expect and require and model compliance by individuals, family leaders, and other communities. And so if we model the behaviors we want to see, sooner or later, those will become the norm. I've included some resources here such that you can go back and use these when this video is made available. These are pictures of coronavirus. This is a detailed picture of the virus. It's an actual picture of the virus. You see the long spikes, we call them, which is why it gets its name corona. There's a crown appearance around the outside. And then this is one view of a portion of an infected cell. Each of these little yellow dots would be one virus that's been produced by this cell. So this just gets you oriented to what we're going to be talking about today. All right, so very quickly, I just want to tell you that coronaviruses are a family of viruses. So we have the viruses that cause the common cold, picornoviruses, or viruses that cause measles, mumps, the paramyxos. They're divided based on their structure and their shape and how they work. So there's a family called coronaviruses. And again, you see in the diagram, these long projections that are called spikes, as you see in the picture here. These are other pictures of viruses. Viruses are sub-microscopy, are sub-life in that they cannot replicate by themselves. They have different shapes. So these are actual pictures. They're very small, so we have to magnify them to high levels in order to see them. So these are just different shapes of viruses, rabies, which looks like a bullet, or Ebola, which looks like a ribbon. And here's again the SARS coronavirus that has caused the pandemic that we're now in the midst of. And then I also want you to understand that all viruses have to reproduce inside of a cell. They're not like bacteria, where if you put bacteria in a water with the right nutrients, it can reproduce all by itself because it's a cell. But viruses are not cells, they're parasites, and they have to get into a cell and go through processes that allow them to make more virus. So what you see here in this photograph are pictures of viruses, coronaviruses, being made in one cell. So in essence, the picture represents the assembly of viruses into new particles that leave that cell to go on. And I also want to remind you of the central dogma you probably learned back in elementary or junior high school, that DNA, which is what our genetic material is, goes to messenger RNA, which is the code that then tells us how to make a protein. We will be talking about this more when we talk about the vaccines. All right, so let's first look at the virus, coronavirus, and let's get some terms straight. Hopefully this will help you to understand a lot of what you're hearing. Coronavirus 2, or what was originally called the novel coronavirus, has now been officially named SARS coronavirus 2. That is the virus. That's what I've been showing you. COVID-19, or coronavirus infectious disease, in 2019 is when it was discovered, is the illness or the disease. So it's like HIV is the virus and AIDS is the disease. And again, I'm showing you images of the virus. This is the very prevalent CDC model. This is coronavirus replicating in a cell. Lots more details than you ever want to know. But the point is it has to get in, put its genetic material in, and then make more of itself using the host machinery. Note that it never goes into the cell nucleus where our DNA is. And then it pushes new virus out. And that's what you see occurring here from an electron micrograph by Elizabeth Fisher of the NIH, where she says this coronavirus is 10,000 times smaller than the width of a human hair. So these are many, many particles being made by one infected cell. All right, so where did coronaviruses come from? I told you that this is a natural biological event, and we call those zoonotic events, meaning that The virus comes from an animal into humans. And we've had lots of zoonotic events. Remember swine flu? You've heard of bird flu. You've probably heard of SARS and MERS. All of these are animal viruses where they don't do any harm to the animal, but they move into humans either through another animal or directly into humans. So SARS coronavirus 2 is one of those. And we've had other coronaviruses. There are hundreds of coronaviruses in different species. It may be that this is not, I'm almost certain that this will not be the last coronavirus that we see adapt to humans. We've had SARS and we've had MERS previously. And so now we have this COVID-19 virus that was declared a world pandemic back in March 11th by the World Health Organization. It has spread all over the world. The definition of a pandemic is multiple epidemics in multiple places, a global pandemic. We have it on all continents. And so this biological adaptation has allowed the zoonotic virus to spread easily from human to human through respiratory spread. And so as of April, remember we started with a small number. As of April, we were up to over 1.93 million cases. And if there are cases, then known they're probably twice or 3 or 4 times that many in actuality. By February 2021, which is where we are now, we've gone up to 110 million confirmed cases around the world of SARS coronavirus infection. And this shows you what's going on in the US. We're not doing so well, or we have not been doing so well, but I'm optimistic that we're turning the tide. This shows you when the first cases appeared, were diagnosed in the US back in March, almost a year ago. And the shutdown of many states, including Michigan, the highest level through the summer of diagnosed cases in July, around 47 to 50,000. And then we hit the holiday period after Halloween and into Thanksgiving and Christmas and New Year's. And so we saw the toll of cases go up because of travel and people coming inside and gathering for the holidays, and also the trailing effect of deaths from people who get COVID and don't make it. So the deaths actually trail the heights, and we see now that the holidays are over, that the numbers are going down, and we see the death tolls going down as well. So this is where we are at the moment in terms of numbers of coronavirus infection. And it's also important for me to let you know that as a country, the US has not done very well. We're probably the leading nation in terms of the number per capita or number of deaths or cases due to COVID as you compare here. And then I also want to bring to your attention that some particular groups, particularly African Americans, are more disproportionately affected by cases shown here. This is for Chicago, data from Chicago. Latinx, African American, white, Asian, other in cases, and the same thing here in deaths. And you see the disproportionate number of deaths or cases. You can see that also in this kind of plot where we have the population that's African Americans here, and the number of deaths as of April. This was April data, so it might be a little different now, but say in Michigan, African Americans are 14% of the population, but as of April 2020, we were 41% of the documented deaths from COVID-19. And there are many reasons for this, including frontline jobs and pay and the need to be out away from home, underlying medical conditions, access or lack of access to medical care and testing, the misinformation that goes around in many neighborhoods. And there are lots of other reasons, many of them from the legacy of enslavement in America. And so I ask you, what can we do about these? And certainly the Fralin Institute and others are very involved in addressing these issues, but we all need to be aware of them. So then how does COVID-19 or coronavirus 2 cause the disease COVID-19? First of all, there are multiple ways that microbes can spread from person to person. And so one can directly contact someone by shaking hands or touching objects such as fomites like this phone or a blanket, or being bitten by an insect or a vector or an injection through the skin. Or in the case which is what works for coronaviruses, or at least for COVID-19 or coronavirus 2, it's in the air, a respiratory virus. So when we speak or sing or talk or breathe, virus that is reproducing in the nasal passages go into the air. And if you're nearby, you can inhale these into your nasal passages and start a new infection. And then there are others that are transmitted by food and water. Fortunately, coronavirus is not transmitted except by respiratory and direct contact with fomites. How easy is this virus transmitted? Very easy, which is why we now have a worldwide pandemic. This is an early example from March, where a person who did not know that he was infected traveled from Singapore to France for a weekend ski trip. And he stayed in a chalet in a cabin with 12 other people. At the end of the weekend, they all went back to their separate countries. And from that has been traced an epidemic that exposed at least 21 people known to coronavirus in 4 different countries, just from one weekend breathing in the same air in this chalet. And so you can see that any one cell, and these are just pieces of a whole cell. The cell would be bigger than my screen, but infection of one cell produces lots and lots of virus. And you can imagine then that when someone is breathing out without a mask, they're putting lots of virus into the air. This is another example of a restaurant in one of the Chinese cities, where the air conditioning system, that there were 2 different rooms with 2 different air conditioning systems. The airflow out and back in here for tables A through F. And then if you blow that up and you look at the people who ended up testing positive for COVID within the next 14 days after visiting the restaurant, we see that A1 was the ground zero person that was asymptomatically infected. The person did not know they were infected. They were there with their family, but there were 2 other families, B and C, who were in the same line of air conditioning. And over the course of the next days, they ended up with positive coronavirus tests. And so B was there 53 minutes and overlap with person at table A, and C 73 minutes. So this was one of the first bits of evidence that this is actually aerosolized or transmitted not only by droplets, but also aerosols. So how do we stop this? To reduce this, the best way is to not be around other people. The next best way is, if you're out for everyone, someone who may be asymptomatically infected or an uninfected person, both of them to wear masks and to keep that distance, the physical distance, such that the droplets fall before they get to the next person. And then if not distance, certainly both people, everyone should mask, both the person producing virus and the person getting infected or that may get infected. And then the other next possibilities with one or the other person wearing a mask. And the worst situation is when nobody wears a mask. So in order to be safe and healthy for our homes and our families and schools and workplaces, we want one of these 3 conditions at the bottom. And that's because virus transmission is highest from asymptomatic or pre-symptomatic people, people who don't know they're producing virus into the atmosphere. So they're not sick enough, they're not even sick, and yet they are producing virus that others can breathe in. All right, so that's the virus. Now, how does coronavirus lead to COVID-19 disease? What does it do to the body? First of all, remember it's a respiratory virus, and people breathe it out into the air anytime they're talking or singing or just breathing or coughing or sneezing without a cover over their mouth. So you can imagine that these droplets and aerosols are going into the air. If you're close enough, you can breathe them into your nasal passages. They bind to the tissue in your nasal passages and your mouth and oral cavity. They reproduce there, travel down the trachea and windpipe into your lungs and into the very small alveoli and bronchioli of the lungs, where they actually reproduce. You can also touch something that these droplets have fallen onto and then touch your mouth or nose or face and transmit them into your own body, self-inoculate. So this is a respiratory transmitted virus. It's one of the easiest ways to transmit viruses. And then I wanted you to understand the timeline from exposure to symptoms and shedding. And this is really, really important. You know that quarantine for someone who's been possibly exposed is 14 days. So if this was the initial exposure, one of the things we realized sort of midway is that virus shedding happens before any symptoms occur, if you're going to get symptoms. And there are people who get no symptoms. So even if I'm going to have symptoms, shortness of breath or a headache or body aches, those may not come until several days after I'm already producing virus and putting it into the environment for other people to breathe. And then the symptoms also only may appear even later. Even so, you're still producing virus early on. So we have clinical infection where there's mild or moderate or severe COVID, or we have subclinical or asymptomatic infection. About 40% of infections have no symptoms, and yet people are still shedding virus. So this is one of the reasons why this virus has become a global pandemic and is so difficult for us to control. Again, you see the example here of the person infected putting particles into the air that are breathed in by an uninfected person who then becomes the person who's infected, and the cycle continues. Once the virus is in the body, the initial symptoms, if they're going to be symptoms, may be fever and headache, muscle pain and tiredness, a dry cough or tickling in the throat. That means the virus is now reproducing on the cells of the nasal passages, and immune defenses kick in. And that's actually part of the issue. This is a battle between the immune system trying to get rid of the foreign object and the virus trying to reproduce itself. So the virus now, there's a battle between the host defense and virus replication in the tissues of the lungs. And depending on who wins this war between our defense system and the virus replication affects what happens to the patient. About 80% of people will have mild to moderate symptoms, so they can handle it at home. It's not fun. It's probably the worst cold or flu you've ever had, but there's no need for additional oxygen. Unfortunately, about 20% of people progress to severe disease, which requires medical attention. And what happens there is that the virus actually gets into the branches and into the small air sacs. And those air sacs are necessary for exchanging oxygen and CO2 to supply energy and allow our tissue, our organs to function. When we get infected, and this is what a healthy lung looks like, the open air sacs, this is an X-ray showing the clear lungs. But when there's infection with COVID-19, there's swelling of the tissue where the virus is reproducing and where the immune system is trying to shut down the virus. There's clogging that decreases the airflow such that you can't get oxygen in or CO2 out. And eventually fluid begins to build up because that's the engineering of the lungs. And so instead of a clear lung that exchanges air, you have a pneumonia fluid-filled lung and the person cannot breathe. And so this leads to not only impaired lung function, but also deprives other organs of the oxygen they need to function. So COVID-19 can lead to a wide range of body effects, from mild things like pink eye and loss of taste for mild situations. And most people actually do have this loss of taste or smell, but also very severe issues, strokes from not having oxygen to the brain or blood clots in the lungs, arrhythmias and heart attacks, kidney damage. Many COVID-19 severe patients go on dialysis, skin issues, purple fingers, purple toes, defective immune systems, GI tract issues, and blood clotting that can affect many parts of the body. And so someone who has severe COVID, the goal is to try to help their organs not fail by supplying external oxygen or keeping them breathing. And so whether somebody survives or not from severe COVID really just depends on whether the organs can be kept functioning. And we've lost over almost 500,000 people in the USA due to COVID-19. For those people who don't die or who survive or who actually recovered, there's something also known as long haulers, COVID-19. And this even happened with people who have what is called mild COVID. In other words, instead of getting over it like you do with the flu or the cold, there are symptoms that linger and linger for months. And even some people who didn't know they were infected find out later that they're COVID positive and they have long hauler syndrome. So brain fog, shortness of breath, these are many of the things that affect people who get infected. Hypertension, heart arrhythmias, joint pain, and these can go on for a very long time. We don't understand the molecular events that are occurring. And so there are many unknown questions with COVID-19 and with coronavirus 2 pathogenesis. How does it cause disease? How does it cause these long-term symptoms? There are treatments like monoclonal antibodies that can help, but we really don't have a way to fix this. So what do we do? What's the best thing to do? The best thing is to prevent the virus from getting into your body or prevent it from replicating in you and traveling to somebody else. And so we've had these all along. We have not done the best that we could with these, but I am hopeful that now that we understand and now that we have another tool, the vaccines in our toolbox, that we will use these tools of physical distancing, staying at least 6 feet away, staying home if we don't have to be out and not gathering, limiting the size of gatherings such that there are fewer people in a room to pick up if somebody is asymptomatic and they're putting virus into the air, correctly wearing our mask when away from home, cleaning surfaces, frequently hand washing. So if we touch a doorknob or elevator button that somebody who is putting out virus has touched with their hand, we don't end up touching our face. Getting tested if potentially we've been exposed, and cooperating with contact tracing and isolation. And then lastly, the research that's been so intense on vaccines and antivirals is very important, along with these public health preventions. And to be honest, the public health preventions are very, very powerful if everyone would use them. This is a reminder before we go into vaccines that wearing a mask is not effective unless you wear it correctly. So this is someone who is putting aerosols with virus into the air, as represented by the red droplets. There are 3 different kinds of masks that have different levels of protection, with the most being the N95, that you can see that it stops the particles, both the aerosols and the droplets, and then the other mask. So know what kind of mask you have, and then wear it correctly. It must cover the nose and the mouth at all times. It shouldn't have a vent. You shouldn't touch the front. And you should wash your hands when you take the mask off by touching the strings and not the front of the mask. So to wear a mask for a little while and let it drop down, like I've seen in the supermarket, below your nose is really not preventing these particles. So it's so important to wear a mask correctly. All right, so in the last part of what I want to tell you about today is we'll talk about the COVID-19 vaccines, which are the newest piece in our toolkit, along with preventions of isolation and masking and keeping distance. Vaccines are really powerful and have changed infectious diseases. So what are vaccines? They are a way to prime the immune response. Our body is wonderfully and fearfully made such that we can fight off something that's not us, not self. And so we've had virus vaccines to smallpox and yellow fever, measles, mumps, rubella, polio, chickenpox, pneumococcus, human papillomavirus, and even now Ebola and COVID-19. So these are very important ways to help control infectious diseases. They protect before infection. So unlike antibiotics or antivirals, you get a vaccine before you see the microbe. One has to decide what kind of vaccine. You can get one that's live and weakened. So you get a whole virus. I'll use my fist as an example, but that is a weakened form, so it can't cause disease. Or you can get a killed virus. Again, it's the whole virus, but it's been destroyed, so it can't reproduce. Or you can get a subunit virus, and that's what the COVID-19 virus is. Instead of putting the whole virus in, represented by my fist, we're just putting in the thumb which is a subunit of the whole virus. And that's also true for the human papillomavirus vaccine or the hepatitis B vaccine. We have to decide when to give it, whether there's once or multiple times, what's the storage, what's the distribution strategy, as you well know here in Virginia. We also look at the benefits to the individual and to the society. We need high uptake if we are to reach a protection level that will protect people who, for whatever reasons, cannot get immunized. And does the vaccine prevent disease, which is what the COVID-19 vaccines that we have so far do, or do they also prevent transmission? And that's being studied now. So in all cases with vaccines or antivirals or drugs, we are weighting the effectiveness, called efficacy, as well as the safety. We want to balance these things. We want to be able to use things that are safe to our bodies, but also work well against the microbe that we're trying to prevent. And then we have questions about, with the vaccines especially, will they be valid and effective still when new variants, new genetic variants that naturally evolve occur? So this shows you a diagram of the COVID-19 virus with the spikes, these regions that I told you that look like a crown. And so the COVID-19 subunit vaccine just puts the spike in, that little thumb that I'm telling you about, using the code, the messenger RNA for the spike, not the whole virus. We have quite a few viruses that are under phase 3 clinical trials, and I'll explain that in a minute. But because of the importance of human trials, we now have very rigid rules about how clinical trials and human research has to be done. So there are 2 virus vaccines that have moved into the emergency use authorization, Pfizer, BioNTech, and Moderna, that are now being rolled out in the USA and some other countries. And then tomorrow, actually, as a member of the FDA advisory panel, we will be having the review of Johnson & Johnson, which is an adenovirus. This is a virus that is not coronavirus, but it actually carries that little thumb, that gene for the thumb on the virus, which is a non-pathogenic, a non-disease causing virus in humans. Whereas these two are just the messenger RNA in a little fat droplet, which I'll show you in a minute. But we have others as well that are in those clinical trials in the large studies, like Oxford AstraZeneca that you've heard about, or Novavax. So what's the clinical trial process? This is a very rigidly implemented plan where experiments get done preclinically in the lab. Then they go into a smaller safety study called a phase 1, where you might have 10 to 25 healthy people to see if it's safe. And then it goes into a phase 2, which is looking at, does it work against the disease to protect you? That usually has more people and can be conducted from 6 months to 2 years. And then we have these large studies, these clinical phase 3 trials, which now up the ante because it's looking at safety. Is it safe for people? And is it effective against the disease? So this is a large scale study, usually typically say for measles, mumps, rubella, or polio vaccines. They were conducted for 5 years or more with about 3,000 people. And then those things that work go into FDA approval. And if they pass FDA approval, they go on to phase 4, which is use and surveillance to look at how we need to adapt those vaccines to make them better, or drugs. So where we are, we have, the numbers are here of the different vaccine formulas that are in the various phases. We have a lot in phase 3, as I just showed you. We have 2 that are authorized in emergency use authorization in the US, and that's Pfizer and Moderna. And I'm going to tell you how that happened. Instead of 3,000 people for 5 years, in order to get sufficient data to know if it's safe and if it works, these vaccines had to have 30,000 people enrolled, and they had to have them in, they couldn't do 5 years because we're in an emergency situation. So they had to be in people for a minimum of 2 weeks or 2 months, and ideally longer. And then for single dose, both of these are 2-dose vaccines. For a single dose vaccine like Johnson & Johnson, they have to have 60,000 people for at least 4 to 8 weeks or 8 weeks in people in order to be able to even qualify to be looked at for an emergency use authorization. And so what is in the COVID-19 messenger RNA vaccines that are approved? Let me explain this. This is very important. So again, we're not putting in the whole virus with all the fingers in my hand, but just the messenger RNA that encodes the spike. And so that messenger RNA, just for the spike protein, is put into a fat droplet called a lipid nanoparticle. It's just a little droplet of oil, and the messenger RNA is protected in that. That material is dehydrated and then shipped to wherever it's going to be used. It's reconstituted with water and buffers and no preservatives, no antibiotics, no adjuvants. And then that is injected into a person's arm. In the body, this nanoparticle, this little fat droplet with the coating of messenger RNA for the spike protein, gets taken up into cells, and using our normal cellular machinery, that messenger RNA is made into spike proteins, viral proteins, not the virus, just the spike protein. It never even gets into our nucleus, so it doesn't even get anywhere near our genetic material. And now, because this is a different protein than our normal protein, our wonderfully made immune system says, oh, this is not me, this is something foreign. And it mounts an immune response, a defense response against these proteins, such that we get B cells and T cells. These are just pieces of our immune defenses that make a memory to the spike, such that when we see the real virus, we say, oh, that's not me. I've seen that before. Let me shut this thing down. And so that's what gives us the protection against the real COVID-19 virus or the SARS coronavirus 2 when we see it. All right, I just briefly want to describe the studies, just so you know that no steps were skipped in getting to the emergency authorization for Pfizer and Moderna. They both had, Moderna had 30,000 people, Pfizer had 42,000 people enrolled in the trials, the clinical phase 3 trials. These were divided into a vaccine group and a placebo group. They both were given 2 injections. They reported their symptoms, which is what the vaccine was looking at. Does it protect against symptoms? People who got symptoms reported them, and they were given a coronavirus test. Those who were positive then were put into the group that, quote, got COVID disease. And when the code was broken for these 200 cases, they had to get up to about 200 cases. The 94 to 95% effectiveness that you hear about comes from realizing or finding that 180 of these 200 people, and I'm just using this as an example, but the numbers are about what they were in the placebo group. They did not get the messenger RNA, they only got the salt and sugar water. Whereas the COVID-19 vaccine that had the messenger RNA with that spike protein expressed, only a few people that got even very mild COVID were in that group. So this says that the vaccine protected against COVID disease at a 94 to 95% effectiveness. And that's what we mean. And now these trials are continuing. So these numbers are going up and they're holding out in terms of protection. People who got mild COVID were in the vaccine group. More people and people who got moderate and severe COVID were in the placebo group that did not mount an immune response. So here are the symptoms that were being looked at. Anyone who showed up with a cough or fever or chills or fatigue and had a positive COVID test were included in those COVID cases. And so we know that the vaccine protects people from the mild to severe disease and death. And that's very, very important because it's the death and the severe illness that's clogging up our medical systems and even preventing people who don't have COVID from getting the medical treatment they need. We know that the vaccine prevents disease symptoms, illness, and death. We don't know yet if it prevents infection or shedding. So anyone who gets a vaccine still has to wear a mask and do the physical distancing and not gathering to make sure that even though they're not getting sick, they're not producing virus and putting it into the atmosphere. And for the last couple of slides, I'm just going to tell you what happens with the vaccines. So these two vaccines are very similar. They're both to the spike protein. One difference is there's a slight difference in the amount of time between the two injections. They both are very effective. They both have some side effects, but those are short-term effects due to your immune response to something foreign, and they go away within 3 to 4 days. They're more severe at the second injection because your body already is primed. So it's saying, oh, I've seen this before, let me attack it. And when you get a fever or body aches after a vaccine, that's a sign of your immune system responding. So when you get vaccinated, you need to, 1, get a date for the second injection, and 2, register with vSafe, where you'll get a text message on your phone to let you report your side effects so that people are keeping up with these side effects. And what we now know at 2 months after the rollout is that the side effects that are actually being seen are very similar or the same as those that were reported in the clinical trial. There's also a site called VAERS, or Vaccine Adverse Event Reporting System, where anyone who gets even more serious things like anaphylactic response, if you're responding to injections, and those are usually people who already know they're allergic to vaccines, they take their epinephrine pen with them, and there are certain procedures they go through, but that can be reported as well. So in summary of the vaccines, Pfizer and Moderna are similar in their both 2 doses. They're both messenger RNA that's putting in the coding region for this spike. They're both effective across genders and races, which were in the clinical trial and now in the rollout. They both are up to 94 to 95% effective. The difference is Pfizer has to be stored at a lower temperature. It can be used down to age 16, and the second dose is given 21 days after the first dose. For Moderna, you can store it at 4 degrees, and it's viable even 30 degrees and 30 days in the fridge. And it's only 18 and up, and the second dose is 28 days. But these are pretty similar in what they do. And the side effects are pretty similar that have been seen. Pain at the site of the injection of the arm, that's pretty typical. That goes away within a couple of days, and you can always take ibuprofen or Aleve or something should you decide to get the vaccine. So all of these are side effects from the immune system actually being built up against this spike protein that's a foreign protein. There are phases of when the vaccine distribution is recommended by CDC, and states have the ability to change this. So we started with the healthcare personnel, with long-term care facility residents, because these were the people that were most vulnerable to death and severe illness. And of course, our healthcare personnel were more exposed to people that might be producing COVID in medical centers. Then 1B are other frontline essential workers like fire persons and others, people over age 75, people 65 with medical conditions and older. And then some states included teachers and school personnel. And then the other divisions are here. So you can find out where you are, and there are people around our state health departments, our county health departments who can help you navigate that. So in summary, these vaccines, the 2 messenger RNA vaccines, Pfizer and Moderna, are approved for emergency use in the USA and several other countries. What they do is they take the coding region, the messenger RNA, with the spike protein, put it into a fat droplet, which delivers it to our cells. The cells recognize it, make the viral protein, put that on our cells and say, this is not me. The immune system then comes in and makes antibodies and memory cells that when you see that spike again on the real virus, you know that it should be destroyed. And so that's the host memory defense that will protect us against COVID-19 disease. And so how do we make this work? Well, there has to be some trust built, that we have confidence in the vaccines, we have confidence in the process, we have transparency. For example, we don't know that these vaccines block infection. We don't know yet if there are any long-term effects, like 3 to 5 years. I wish I could say that we know, but we do not know. We have to know that there's inclusion of diverse populations, that there's equitable access, and that has been a real big issue. that there's community engagement and investment in this process, which we see in many areas of the Virginia and other states. And then we have to have trusted messengers, that people who we can ask questions about and be sure that we're getting transparent, clear answers, that nothing's being hidden or kept away from us, such that the goal is to have high buy-in. It's necessary for most people to become immune either from the disease, surviving the disease, or getting an effective vaccine in order that we can shut down the virus replication. We need to shut down the virus replication because the more the virus reproduces, the more opportunity it has to change and adapt to get around our immune systems and make new virus variants. And that's the danger of allowing COVID-19 to continue, because more coronavirus is getting produced, and the more that gets produced, the more opportunity there is for it to mutate in ways that will help it. So what do I want to tell you? How to stay safe? You need to make a plan, remain with your immediate family, use the 3 W's, wear your mask, wash your hands, watch your distance. If you have symptoms or think you've ever been in contact with someone, get tested, do the contact tracing cooperatively, trust but verify information when it comes to vaccines, decide what you need to do for you and your family, consult your health provider, and with or without a vaccine, always be proactive to reduce the coronavirus spread so we can get ourselves out of this pandemic. And so the takeaways, which is what I gave you at the beginning, that the pandemics are a part of just nature adapting. And we need to shut down the replication so we don't have more adaptation of variant strains. For COVID vaccine, you need to decide, make an informed decision. But these are highly effective, amazing science that has produced vaccines that protect us against COVID disease. And then lastly, make and implement a plan to prevent infection, whether you have a vaccine or not. These things we're going to need to do until the level of testing positive is 3% or lower in a community. We are really all in this together. It won't work unless we all get on board to do what we have to do. So continue the conversations. I thank you for allowing me to be a trusted messenger today. These are just some resources. And I think we have some time for questions, and I'll just stop here and turn it back to Dr.
Thank you very much, Dr. Fuller. It's fascinating, extremely informative. I want to first report on the poll that you ran.
Sure.
Of the respondents, 85% said yes, they would take the vaccine.
Great.
6% said no, and 9% were not sure.
Wow. Great. Thank you.
Yes, that's good to see. So we do have a bunch of questions. Let me try to select a few for you and read them. The first one is from one of our researchers here, a postdoctoral fellow, Dr. Monet Roberts, and she said, "Thank you for the amazing talk. As a Black scientist, I often engage with my family members and friends, and the distrust in the general Black community, as you referred to earlier in the presentation, is still very apparent today because of illegal practices done historically. For instance, I've been trying to convince some of my family members and friends to get vaccinated, and they are resistant. How do you suggest we engage with those within our community to get them to gain trust in science and medicine?"
Yes, that's a question I get often. And I think you have to meet people where they are. You have to acknowledge that you have a right to question this. But let's talk about the questions that you have. The things that have happened historically, we need to know about them to make sure we don't repeat them. But there are guidelines like the clinical trial practice, the internal review boards for human studies, and really then to help them talk about things that they're very unhappy about, or not unhappy, but unknowing about. The other piece is to tell people that, I know you really want to get together for Thanksgiving. We really want to have everybody together for Christmas. And we can't do that until we are all safe from this. So one of the things that every person can do is to help this process by agreeing to get the vaccine, or if not, then having to absolutely follow all the preventions until there's no more virus. And we think this virus is probably going to be around at some level. So meeting them where they are, and then using the things that emotionally appeal to them. Most grandparents or older persons or even younger persons want to be with family. And so you say you can be with your grandkids, we can go to the movies eventually if we all do this. It's a hard thing, but I think if you do it for the right reasons and don't shame them or blame them, it really can make a difference over time. And make the appointment for them and say, you don't have to keep it, but you got it.
Okay, thank you. Karine Calivras says, I'm curious about the ethics surrounding the continuation of vaccine clinical trials if the vaccine has been shown to be 95% effective. Are those who received the placebo previously now getting vaccinated? And if so, how can the long-term effectiveness be studied? Sounds like an ethical dilemma.
It is, and it was really discussed at length at the FDA panels. And I must say, I don't know what the conclusion was as to how those companies were advised to go forth, but the goal was to keep the study going. But as a very astute question person said, if you know the vaccine works, is it not ethical not to give them to them? So what the last I understood is the proposal was to do a crossover study where after a certain time, those who got the placebo got the vaccine and those who got the vaccine got the placebo, and keep everyone in the study to look at how long the immunity lasts and other things. But yes, he is absolutely correct. It does bring up a dilemma for continuing the studies. These studies are supposed to go for 2 years, by the way, and these companies have to still apply for licensing. This is only emergency use authorization, not a license for the vaccines.
Okay, thank you. Chris asks, what has been the impact on the homeless population? What can we estimate about their death rate?
I don't know the answer to that in terms of death rate. I do know there's been a lot of attention to making sure that the homeless population have access to food and vaccine and have distancing options that can help keep them safe, and masks and those sorts of things. So, but I don't know the answer to that question in terms of stats.
Okay. One of our attendees had several questions for you. I think it's somebody you know named Gerald Saunders. Said he's a zoology graduate from UNC Chapel Hill. They're thrilled at your accomplishments. They remember you from campus. So his question is, can we use so-called vaccines as such, or are they just pharmacologic therapies?
These are real vaccines, as in something that prevent disease. The difference between them and measles and mumps and polio and smallpox is we just don't have the history of knowing the long-term effects, but they are real vaccines. They are preventing disease.
Okay. Scott Winter asks, how many people have died of COVID versus people who have died with COVID?
I don't know the answer to that because of COVID. So, for example, I share this not for your. I appreciate your sympathy, but my mother passed in May of this past year, not of COVID. She did not have COVID, but she had diabetes for many years and just wore her body out. So she was home with us, and therefore, I don't think things could have been different. But did COVID affect her death? She was not recorded as a COVID death, right? And so there are lots of examples where people are affected by the fact that our hospitals and medical centers are having to go through amazing procedures because of the presence of coronavirus. And so I can't answer that number. I know people who have died with COVID, at least that we know about, are around 500,000 just in the USA. 500,000, I believe, is the number that we're just hitting about now. And that's too many, too many. We could have changed that. We need to change that.
Okay, thank you. Robert Brown asks about brain cognitive fog. What is the pathophysiologic basis? Is it vascular, direct cytotoxicity? And also asks if the virus crosses the blood-brain barrier.
I wish I knew what the pathogenesis of brain fog is, and I don't know if anybody does. There might be. There's so much research going on right now to understand molecularly and mechanistically what goes on. What does coronavirus do to the body that allows all this wide range of symptoms that we're seeing to occur? And so there's some thought that it affects the initial innate, the early immunity to sort of circumvent the immune system from the beginning, those things that should shut down viruses or bacteria. And so what else happens, we don't know. There are all kinds of studies, the blood clotting that it causes. So there are other people that know that. I don't know that, and I would love to know myself. Great questions. I wish I had answers, but I don't.
Okay. We have an anonymous question. They say it used to be vaccines were associated with universities, for example, polio in the University of Pittsburgh. Why has that shifted to drug companies? Is there any university involvement?
There's lots of university involvement. And that's a great way to segue into one question that's often asked is, how did we get these vaccines so quickly? Coronavirus just evolved to be in humans that we knew of in November of 2019, a year ago. And now a year later, we have two vaccines and more. So what happened is, particularly the messenger RNA vaccines are not new technology that was discovered from, ground zero to emergency use. There were people that have been working on this technology for 10 years or more, 12 years or more. And messenger RNA technology was used with cancer therapy. So what happened is researchers from the NIH, another African-American woman, Kissimmee Corbett, and others, as well as other researchers from universities, from the pharmacy companies, worked together. I describe it, we were all making pieces of quilts. And instead of completing your piece and your quilt by yourself, you now put your piece together with other people, and soon you can get to the full quilt. So the cooperation of the scientific community to get to these vaccines that are safe, as we know, and effective, as we've seen, against disease is pretty amazing technology.
Okay, there are still a few more questions. How about I'll give you two more and then we'll wrap it up, okay?
Okay.
One is from a professor here at Virginia Tech who's head of our public health program, Dr. Laura Hungerford, and she asked, who do you think would be the best trusted messengers for helping communities make the best decisions for dealing with COVID?
I don't know if there is a best trusted messenger. I think it depends on who the message is going to. So clearly our religious leaders, but our politicians, our granddaughters and grandsons who, want to be able to go get grandmom's mac and cheese. I think the message really has to go out from everyone who understands it. And I think we need to use multiple people, whoever will share with somebody. And I think every one of us really is responsible for helping somebody else. There are many people who cannot easily navigate the computer systems necessary to sign up to be in the vaccine registration line. So people can help do that. So I don't think there's a magic formula here. We are all in this together. If we don't get high uptake of either those preventions that we've known about and not done great with, and or the vaccines, which are protective against disease, and we hope protective against shedding and infection, we'll know, I'm sure in a couple months, because those studies are in progress. We need everybody to do this to get out of it. And not just for the US, but this is a global pandemic, right? So if we've done it here, but they have not done it in Zambia, then we still have the threat of the virus changing to something that's going to start it all over again. We're in this thing together.
Okay, thank you. We'll take one final question. It's also from an anonymous attendee. They ask, is it safe for two fully vaccinated individuals who live in different households to interact without practicing social distancing guidelines, or should they still adhere to them?
Two fully vaccinated people, if there's nobody around but those two people, could interact because they're protected against disease, right? But they should not be around anybody who's not vaccinated, because either of them may be shedding virus, and that could go to the other person. So at some point, we hopefully get to that point where we are like that. We're around people who are vaccinated, and we can relax those preventions a little bit. But until we're there, the right thing to do is to protect the person who is not yet vaccinated, who does not have an immune defense. And the last thing I want to say, Dr. Friedlander, is that hopefully 3 to 5 years from now, everyone is going to be immune to coronavirus that we have now. And either that's going to be from getting the virus infection, whether asymptomatic or with disease, surviving it, no matter what that cost or what that felt like, and now having some natural immunity. And we know that the COVID vaccines are better defenders. The defense is stronger than the natural immunity. Or people will get the vaccine by this little spike, or maybe choose another one of the vaccines if they're approved and get immunity from a vaccine. But people will have immunity one way or another. And we just think that getting the vaccine, even though there's still a few unknowns, it's not going to change your DNA. It's not going to make you have 2 heads or 3 hands or any of those conspiracy things that are out there. That's the better way, the stronger way, and the safer way, less risky way of getting that immunity. So I hope that helps people who are trying to figure out, will I do this? Based on the poll, 85% people said yes. That's the highest I've seen so far in a while.
Oh, that's good. Good to know for our community. Well, Dr. Fuller, it's been extremely informative. We appreciate the straight talk and the optimistic note at the end, but based on science and fact. Thank you again very much. Please join me in thanking Dr. Fuller for a wonderful presentation. Thank you so much. That was great, Oveta. Thank you. Thank you so much.
Thank you for the invitation. I hope it hit home for folks.
Yeah, very good. A lot of really good questions. I'm glad to hear our community had the highest positive response rate you've seen so far. That's great.
It was 83 at another place about 2 weeks ago at Michigan State University, but actually in communities of color, it's typically 60, 50. It's going up some, but not at that 85%. So that was good.
Yeah, great. Well, look, thanks again. I'll see you in a little while, okay?
All right.
All
Take
right.
care.
Thanks again. Take
Thank
care.
you,
Bye-bye.
Bradley.